Hello students, this is Dr. Faizan Mirza. Here in this video, I'm discussing with you the biosynthetic pathway of GA1 gibberellin, active gibberellin, and how the alleles LE and small LE and the enzyme GA3 beta hydroxylase actually help in destruction of this DELA protein that leads to growth in plants. So biosynthetic pathway, I'm discussing this with you. So we know that active gibberellin is required for elongation of plants or growth of plants. So this is called GA1. GA1 is the active form of gibberellin. It allows growth by mitosis. So wherever you use it on GA1 is active gibberellin. This wherever it's present, this GA1 will end up producing stem elongation. GA1 gibberellin is a plant hormone. This GA1 is produced by a precursor molecule called GA20. So plants which actually grow tall, they have GA20, which converts to GA1 through an enzyme called GA3 beta hydroxylase. Plants who stay dwarf, they also have the same precursor GA20, but GA1 is not produced because the enzyme is non-functional. Why is the enzyme non-functional? Because the non-functional enzyme is having the amino acid threonine at its active site. The enzyme threonine present at the active site of this enzyme fails to catalyze this reaction. But if at the same active site alanine enzyme was there on the active site, the enzyme is very well working. It allows GA20 precursor to be converted to GA1. So you know the enzyme name, how it is converting the precursor molecule to active gibberellin. You know the change in the amino acid. This is an example of missense mutation where a codon changed in that particular allele and the amino acid changed as well. We know when the amino acid changes, it changes the 3D shape of the protein, and the changed protein then cannot work in the same way as the actual protein was working. Now, since this is we are discussing this to the genetic level, so what is the genotype here? The genotype is capital LE, or capital LE, small le. At least a single dominant allele of LE is required for this working enzyme to be there. So if a plant is having capital Le dash means this capital Le could be capital Le, or it could be capital Le with a small le. Both cases will have at least one dominant Le allele there. This allele produces the enzyme having alanine at the active site. This produces the enzyme GA3 beta hydroxylase, which allows GA20 to be converted to GA1. And if the allele is homozygous recessive for both small le, the plant stays dwarf. Why? Because the enzyme is non-functional. It is having threonine at its active site. This fails to catalyze GA20 to GA1, so no active gibberellin forms. When GA1 is not forming, how this affects growth? So let's go to the to gene control in eukaryotes, how GA1 actually promotes growth. So once this GA1 is there, what it will do. So we are discussing the gene control in eukaryotes. So let's recall there is a DELA protein, which is a regulatory protein. This DELA regulatory protein holds or is bonded to transcription factor PIF. This transcription factor, if it is forming a complex with DELA, it cannot come and attach to the promoter. When this transcription PIF cannot attach to the promoter, growth genes cannot be transcribed. So for growth genes to be transcribed, you have to allow PIF to come and attach to this promoter here. We know that in eukaryote, the promoter must allow attachment of transcription factor together with RNA polymerase. So if both will attach, only then the gene will start to get transcribed. So DELA is an example of a regulatory protein that prevents PIF from binding to the promoter region. So where the GA1 comes, if this GA1 is produced according to the pathway that I've just discussed shortly, earlier in this video. So GA1 is produced. This GA1 will bind to the receptor. Together with the enzyme, it will form a complex of gibberellin, GA1, the receptor, and the enzyme. Together with they will cause destruction of DELA. Once DELA has been destructed, this PIF is now free to attach to the promoter region. It bonds to the promoter region, it binds to the promoter region, it attaches here. When it attaches here, transcription of growth genes start. These growth genes are called XET genes. These XET genes will be on the DNA will get transcribed to mRNA. They will allow those proteins to be produced that allow stem elongation to occur in plants. So this is how the stem elongation produces continues in plants. This GA1 is not the same as the gibberellin, which allows seed germination to occur. That was in chemical communication. There was another gibberellin there that allowed the seed germination to occur. We studied that this germination, when seed is to germinate, water is taken up by the seed. This water actually allow the embryo to produce gibberellin. This gibberellin is not the GA1 gibberellin, but another form of gibberellin. This gibberellin acts on the aileron layer. The aileron layer then allow the gene transcription to occur, and amylase enzyme is produced into the endosperm. This amylase will go and start to digest the starch into maltose in the endosperm, and the maltose is then used in growth and the growth in respiration. So this allows the seed to germinate. In the same chapter earlier, we have discussed the role of how the stomatal closure and stomatal opening works. So we can just recall that as well. So in the previous chapter, homeostasis, we discussed the stomatal opening, that how the protons they move out of the guard cells when they go out by active pumping. Potassium ions coming, nitrate ions come, chloride ions enter, the channels open, they reduce the water potential inside, and the water moves in as well. When water moves in, the cell becomes like swollen, it becomes turgid. When the cell becomes turgid, the stomata opens. Now, this occurs, stomatal opening occurs when there's an increased light intensity around the plant, or there's a low carbon dioxide concentration in the air spaces within the leaf. What about the stomatal closure? The stomatal closure is guided by the enzyme abscisic acid, or sorry, hormone abscisic acid, not enzyme, the hormone abscisic acid, ABA. The hormone abscisic acid binds to the receptors on the cell surface membrane of the guard cell. This prevents the H plus from going out. This also stimulates the opening of calcium channels. These calcium channels on the cell membrane and tonoplast acts as calcium to enter. Once calcium enters, it acts as a second messenger, which actually was involved in preventing the H plus from going out. So once H plus stays in, the channels open that allow negative ion and potassium to move out. Water also moves out by osmosis. Guard cell becomes flaccid and the stomata closes. What are the conditions for stomata closure? Darkness, high CO2 in air spaces, low humidity, high temperature, and water stress. We can also recall the electrical communication in plants. And electrical communication, we have discussed the venous flytrap, how the venous flytrap works. The venous flytrap is having two lobes. There are three sensory hairs present on each lobe. There are stiff hairs present on the outer edges that can interlock. So when the stimulus is on the sensory hair, which are three on each lobe, two hair are deflected within 20 to 35 seconds, a very small time zone. Action potentials are generated. These are electrical signals of depolarization. The midrib is having hinge cells. These hinge cells allow H plus to move into the cell wall. This H plus outside the cell wall, they break the hydrogen bond, the cross links between the cell wall, and the cell wall weakens. The middle lamella dissolves. The middle lamella dissolve allow calciums to enter into the cell. Calcium entry to the cell triggers water to move in as well. By osmosis, these cells become turgid. When these cells become turgid, the lobes become swollen or biconcave, and the traps shut very fast. When the trap shuts very fast, the stiff hair on the outer edges they interlock. The fluttering of insect keeps the trap shut. The insect is digested. Once the insect has been killed, the fluttering will stop and the traps will open. We also discussed auxin, the auxins which are indole 3-acetic acid, which actually allow the plant cells to grow in size, not by mitosis but by growing in size. They don't trigger cell division; they allow the cell to grow in size. We do not have apical dominance syllabus anymore. So how auxins work? The auxins also bind to the receptors, the protein receptor on the cell surface membrane. They allow the proton pump to start pumping H plus ion outside the cell. This breaks the hydrogen bond between the cellulose cell wall. This weakens the cell wall. A channel for potassium opens, allowing the potassium ions to enter into the cell. This lowers the water potential inside the cell, and water starts to enter into the cell, causing the cell to become swollen. Now, since the cell wall is weakened, there are these H plus ion also activate proteins called expansins. These expansins proteins are there inside the cell wall, and these expansion proteins allow stretching of this particular cell wall. So the water keeps entering and the cell keeps increasing in size. This is how auxin keeps working. Once the auxin stops being attached here to the receptor, all the process will reverse and will rather will stop, and further entry of water will be stopped, and the hydrogen bond will reform at the surface of the cell wall between the cellulose microfibrils. I hope I tried to cover all the plant hormones in our course here. I hope this was helpful for you. That's it from my side. Thank you so much.